Hello everyone, welcome back. Today I'm here to discuss about the cloning vectors which are used for E. coli. Like we have plasmids, bacteriophages, cosmids and BACs. So I will try to discuss about these cloning vectors in this presentation. Let's start. What is vector? That I have already discussed, that any mobile element of DNA that can carry a foreign fragment of DNA into the host cell is known as vector or vehicle. तो ऐसा मोबाइल डीएनए का एलिमेंट जो कि फॉरेन डीएनए के फ्रेगमेंट को एक होस्ट सेल के अंदर ले जा सकता है और उसे वहाँ पे मल्टीप्लाई कर सकता है, क्लोन कर सकता है, दैट इज नोन एज वेक्टर और व्हीकल. So there are various vectors which are used in recombinant DNA technology on the basis of insert size. So there are different types of vector, like we have plasmid vector, bacteriophage vector, then we have cosmids, phagemids, YACs, BACs. तो different तरह के हम vectors use करते हैं इस technology में. And on the basis of insert size means कितना foreign DNA का fragment हम उस vector में carry कर सकते हैं. तो on the basis of that there are different types of plasmid vectors, like we have plasmid. तो जो plasmid होता है, it is the extrachromosomal DNA which is present in some eukaryotes bacteria, जिसको हम as a vector use करते हैं. और इसमें हम जो insert size है, that is less than 10 किलो बेसिस का डीएनए हम इसमें अटैच कर सकते हैं और इसको मल्टीप्लाई कर सकते हैं इनसाइड द होस्ट सेल, दैट इज E. coli for example. The next is bacteriophage. Again, it is a virus which infects bacteria, so it can also carry a insert size of 9 to 15 kb. Then cosmid, इसमें जो insert size है, that is larger as compared to plasmid and bacteriophage, so it can carry a insert size of DNA that is 23 to 45 kilo bases. 45 किलो बेसिस तक का डीएनए ये इंसर्ट ये कैरी कर सकता है और उसको मल्टीप्लाई कर सकता है. दैन देयर आर सम वेक्टर्स विच कैन कैरी अ लार्ज फ्रेगमेंट ऑफ डीएनए फॉर क्लोनिंग, दैट इज बैक, दैट इज बैक्टीरियल आर्टिफिशियल क्रोमोसोम्स, जो कैरी कर सकते हैं डीएनए अप टू 300 किलो बेसिस या इससे कम. दैन वी हैव बैक्स, दैट इज phages artificial chromosomes, phage derived artificial chromosomes. They can carry the size of DNA insert of 100 to 300 kilobases. Then we have YACs, that is yeast artificial chromosomes. ये carry कर सकते हैं insert size of 100 to 3000 kilobases और minimum 1000 kilobases तक हम इसमें DNA का insert जो fragment है उसे insert कर सकते हैं. Recently there is scientists have developed another type of vector that is called MAC, that is mammalian artificial chromosome. तो इसको बनाने की एक experimentally कोशिश की गई है. So these are some various types of vectors that are used in recombinant DNA technology. और बहुत सारे factors involve होते हैं कि हमने कौन सा plasmid use करना है. So there are not a lot of factors which decides कि कौन सा vector use करना है. So it includes factors like size of insert copy, मीन्स कितना साइज़ होना चाहिए डीएनए का, उसके बेसिस पे डिपेंड करता है कौन सा वेक्टर यूज़ करना है. देन नंबर ऑफ वेक्टर्स मीन्स विच अफेक्ट्स द यील्ड ऑफ रिकॉम्बिनेंट डीएनए, कितने वेक्टर्स प्रेजेंट होते हैं एक सेल के अंदर, दैट आल्सो अफेक्ट्स द क्लोनिंग ऑफ डीएनए. देन द मेथड यूज फॉर क्लोनिंग, कि मेथड कौन सा यूज़ किया जा रहा है क्लोनिंग के लिए. सो दीज फैक्टर्स आर कंसिडर्ड वाइल सेलेक्टिंग अ क्लोनिंग वेक्टर. तो क्लोनिंग वेक्टर को सेलेक्ट करते हुए हम ये कुछ फैक्टर्स को स्टडी करते हैं before selecting any of the vector. If the size of insert DNA is exceptionally large, then we have to use some special vectors, like we have BAC and YAC vectors. तो इन्हें mostly use किया जाता है क्योंकि इसमें जो insert size होता है वो काफ़ी बड़े size का होता है. So mostly जिसमें हम whole genome decipher करते हैं, means जो बड़े बड़े genome projects हैं उनमें इन vectors को use किया जाता है. Then most commonly used जो vector है in simple experiments recombinant DNA technology में, that is plasmid. And as we all know that plasmid is the extrachromosomal DNA which is present in some bacteria. So this is extrachromosomal DNA molecule naturally present in bacterial population, like we have E. coli, and carry genes for some important traits like antibiotic resistance. तो ये बैक्टीरिया में genes कैरी करता है for some antibiotic resistance, जिससे antibiotics का इफेक्ट इन पे नहीं होता है. Then each plasmid carries an origin of replication. So these are the properties that I have already discussed, and thus replicates autonomously within the bacterial cell without affecting its normal replication. So firstly, I will discuss about the cloning vectors which are used for cloning inside the E.coli. And the most commonly used vector is plasmid. और plasmid होता है extrachromosomal DNA that is present inside the bacterial cell, especially in case of E.coli. तो इसमें जो extra chromosomal DNA present होता है that is a circular double-stranded DNA which is known as plasmid. और इसमें जो size होता है plasmid का that ranges in between 1 to 300 kilobases. तो इतना ही साइज का DNA प्रेजेंट होता है प्लाज्मिड में और इसे हम यूज़ करते हैं एज अ क्लोनिंग व्हीकल. Firstly, plasmid vectors. So these are basically extra chromosomal DNA which is present in the bacterial cell, and it contains many genes which provide resistance against the antibiotic. So the most commonly used vector or plasmid is PBR322, and this was the first vector used in practice, which were commonly cloning vectors in E. coli, and its name is derived from that. Here P stands for plasmid and BR stands for the name of its constructor, that is the Bolivar and Rodriguez, and 322 is a number which helps in differentiation of other plasmids from this plasmid. So this is a number which helps in differentiation from other plasmids. So the plasmid from other develops in the same laboratory. To differentiate those plasmids from this plasmid, this number was assigned to this plasmid, that is PBR322, and this is the most commonly used plasmid, and it contains various genes. It contains ORI, that is for origin of replication, then it contains various restriction sites for different enzymes, and it contains genes for antibiotic resistance. So basically this plasmid is a double-stranded circle which contain 4361 base pairs. तो इतने base pairs इसमें present होते हैं, and it maintains high copy number, that is एक ही cell के अंदर इसके 20 से 30 copies present हो सकते हैं. So it maintains high copy number, and it contains genes for resistance to ampicillin and tetracycline. तो ये दो genes present होती है. AMPR resistant means this is gene for this antibiotic resistance, ampicillin, and this is gene for tetracycline resistance, and the molecular weight of this plasmid is 2.83 into 10 raised to 6 daltons. So this is the basic information regarding this plasmid, that is PBR322. Next, this vector contains the restriction sites which are cleaved by some specific restriction enzymes, like we have this EcoR1, PST1, then HIND2, BAM1, SAL1, and CL1. These restriction enzymes are derived from various bacteria, like we have this PST1 which is derived from bacteria Providencia stewarti, then this BAM, it is derived from bacteria Bacillus amyloliquefaciens, and SAL1 that is derived from bacteria Streptomyces albus, and last one CL1, it is derived from the bacteria Caryophanon latum. So ये bacterias हैं जिनसे ये restriction enzymes isolate किए गए हैं. They cut this plasmid from a particular portion. Next, this PBR322 is genetically engineered DNA which is derived from 3 different types of plasmids, like we have this R1, R621, and PMB1. तो इन 3 plasmids की help से ये जो most commonly occurring और used plasmid है, इसे genetically engineered किया गया था. Here R1 and R6 ये जो plasmids हैं ये derive किए गए थे एक बैक्टीरिया से जिसका नाम है Salmonella paratyphi. So these were derived from one bacteria named as Salmonella paratyphi. Species name is paratyphi. तो इससे derive किया गया है ये plasmids which are R1, R6 to 5, and third plasmid used was PMB1. Here जो antibiotic resistant genes है, that is ampicillin resistance and tetracycline resistance, these parts or these genes are derived from these plasmid, that is R1 and R6 to 5, while this origin of replication was derived from this plasmid, that is PMB1. So ये जो different portions हैं, ये different plasmid से derive किए गए हैं. So antibiotic resistance genes for ampicillin resistance and tetracycline resistance are derived from these plasmid R1 and R6 to 5 respectively, while this origin of replication is derived from PMB1 plasmid. So this is basically, this PBR322 was basically a combination of these three plasmids. And this PMB1 is previously isolated from bacteria, that is Bifidobacterium longum, and has been characterized by physical mapping. तो इस बैक्टीरिया से सबसे पहले इसे आइसोलेट किया गया है, that is Bifidobacterium longum. Next vector, which was constructed by certain modifications in PBR322, जो PBR322 है उसके फीचर्स को मॉडिफाई किया गया और उससे एक और बेटर प्लाज्मिड बनाया गया जिसका नाम है PUC19. Why we call it as PUC19? Here P stands for plasmid and UC stands for University of California. So this was engineered in University of California, so that's why this is named as PUC. 19 is the number assigned for its differentiation from other vectors. So PBR322 was modified into PUC plasmid, that is because it is produced in University of California, that's why it is named as PUC. It is 2686 base pair long, so it means इसमें जो plasmid में PBR322 में जो non-essential part था DNA का, that was removed. So it contains unique cloning sites for multiple restriction nucleases and ampicillin and resistance genes to permit identification of transformed cells. तो इस ampicillin resistance gene से हम differentiation कर सकते हैं, identification कर सकते हैं transformed cells की, because जो culture medium जिसमें हम cells को डालेंगे, that contains the genes for this ampicillin resistance, those cells will survive and the cells which lack this gene will be vanished out. So selection of recombinant is achieved by insertion and activation of components. That is the presence of reporter gene, which is mostly LAC-Z gene, which codes for enzyme beta-galactosidase gene. तो इसको inactivate किया जाता है और इसे हम selection में help करते हैं. A complementary portion of this gene being provided by using specially modified E.coli host cell. तो इस complementary portion gene का ऐड किया जाता है जिससे ये जो जीन है वो इनएक्टिवेट हो जाती है. स्मॉलर प्लाज्मिड कैन बी यूज टू कैरी लॉन्गर सेगमेंट ऑफ फॉरेन डीएनए. तो मोस्टली हम स्मॉलर जो फ्रेगमेंट्स हैं, स्मॉलर प्लाज्मिड्स हैं, उनको यूज़ करते हैं ताकि उसमें ज़्यादा से ज़्यादा जो जीन ऑफ इंटरेस्ट है उसको इंसर्ट किया जाए एज कम्पेयर टू नॉन एसेंशियल डीएनए विच इज़ प्रेजेंट इन द प्लाज्मिड. सो मोस्टली हम इसे यूज़ करते हैं. एंड दिस डिक्रीज इन साइज इज़ यूजुअली एसोसिएटेड विद द एलिमिनेशन ऑफ सर्टेन नॉन एसेंशियल जीन्स. तो जो ये साइज छोटा होता है प्लाज्मिड का, इट मीन्स कि जो नॉन एसेंशियल जीन्स प्रेजेंट होती हैं वो यहाँ से एलिमिनेट हो जाती हैं. So that's why there is decrease in size of this plasmid, and this plasmid becomes more efficient in copying the gene of interest. So it helps in elimination of certain non-essential genes that leads to reduction in size of plasmid. Then increase in the number of plasmid copies per cell, जिससे जो number of copies है plasmid की एक cell के अंदर वो increase हो जाएगा number. A single cell can carry multiple copies of this plasmid of smaller size. And also by modification of sequences of PMB1 replicon, the copy number of plasmid may be increased to 500 to 700 cells. अगर हम इस PMB1 replicon का जो हमने sequences लिए हैं इस vectors, इस plasmid से अगर हम इन्हें modify करते हैं तो जो plasmid है, plasmid का number है per cell that can be increased up to 500 to 700 per cell. So इतने plasmids एक ही cell के अंदर present हो सकते हैं. So this cell has very high copy number of plasmid and it also helps in cloning of gene within a single cell. The next cloning vectors which are used in case of bacteria are bacterial artificial chromosome vectors, BACs. So they can carry a DNA insert size of up to 300 or less than 300 kilobases. तो इतने साइज का फॉरेन डीएनए कैरी कर सकता है. and this vector was developed for cloning and stable maintenance of large DNA fragment that is up to 300 kilobases a piece of DNA in E. coli. तो इतने insert size का DNA ये E. coli के अंदर cloning कर सकता है, and it contains ORI, that is for origin of replication. Then it contains certain genes which helps in segregation of BAC only between daughter cells, that is PAR genes. Then it contains LAC-Z gene that I have already discussed. So this gene helps in detection of transformed and non-transformed cells. Then it contains one antibiotic resistant gene, that is CMR. So this antibiotic gene also helps in differentiation of transformed cells from non-transformed cells. So popularly, these type of vectors are basically used for whole genome mapping. जो large प्रोजेक्ट्स हैं जिसमें बड़े बड़े जीनोम्स की सीक्वेंसिंग की जाती है, उस प्रोजेक्ट्स में हम इन वेक्टर्स को यूज़ करते हैं, लाइक फॉर द सीक्वेंसिंग ऑफ जीनोम्स ऑफ माइक्रोब्स, प्लांट्स एंड एनिमल्स. And the copy number of these cloning vector is rigorously maintained at 1 by BAC replication system derived from E. coli, that is F plasmid. F is for fertility factor. So इसमें जो copy number है वो एक ही maintain किया जाता है एक cell के अंदर, and this was derived from this F plasmid of E. coli. Then it is used in host cell which are recombination deficient. ऐसे host cell जिसमें recombination जो RDNA है उसे हम insert नहीं कर पाते हैं, means it fails to carry this RDNA, and allows DNA that is unstable in higher copy number. और ऐसे host में जिसमें higher copy number maintain नहीं होता है recombinant DNA का, तो उस cloning system में हम इसको use कर सकते हैं BAC को without any deletion and rearrangements in the DNA. तो stably हम large size के DNA के insert को we can clone by using this vector. And this is used for sequencing of several organisms with large genome size, including human, mouse, and model plant, that is Arabidopsis thaliana and rice. तो इनके genome की sequencing के लिए इस vector को use किया गया है. The next important bacterial cloning vehicle is bacteriophage vector. The bacteriophage is basically a virus which infects bacteria. So we can ligate the gene of interest in virus genome. Virus के DNA के साथ हम इसे ligate करते हैं, and it can be efficiently inserted inside the bacterial cell for its multiplication. So this is the easiest way to ligate gene of interest in viral genome, and after that this virus will infect the bacterial cell with higher efficiency and can produce has higher frequency of production of cloned DNA, that is this desired fragment of DNA. तो इसकी बहुत सारी copies ये host cell के अंदर produce कर सकता है. And the most frequently used bacteriophage for this purpose is M13 phage. Here this M word is derived from the city Munich. This is the city in Germany. So this bacteriophage was first discovered in the wastewater of this city Munich. That's why this is named as M13 phage. So this is a vector used for cloning of DNA, especially used for sequencing the inserted DNA. तो इसे हम mostly use करते हैं, and this bacteriophage contains genes which codes for different proteins, so they can be grouped in different groups. Like we have this one, that is phage DNA genes, जो कि DNA की synthesis में help करती हैं. There are genes like we have 2, 5 and 10. Then there are genes which are responsible for formation of this proteinaceous cover, that is called capsid. तो इसके लिए different group of genes हैं. Then assembly के लिए different group of genes हैं. So there is division of labor that is shown by the different genes. जो ये वायरस है basically it is a filamentous virus, और इसका जो genome size है that is 6.4 kb. So ये इसका genome size है, और इसमें जो DNA होता है that is single standard circle of DNA. This is very important feature of this bacteriophage. जो M13 होता है, it enters the E. coli through bacterial sex pilus. Pilus means this is a correction, एक bridge की तरह structure होता है ये दो bacteria cell के बीच में, जिससे ये DNA को exchange कर सकते हैं. So it can enter from the sex pilus from one cell to another bacterial cell. A single stranded DNA in virus particle is replicated through an intermediate circular double-stranded structure which is known as replicative form. इसे कहा जाता है replicative form. तो इस portion से ये replication शुरू करता है. And it contains two strand, that is plus strand and minus strand. और जो future progeny वाला virus होता है उसमें सिर्फ plus strand होता है. So only the plus strand is packaged into the new virus particle. The most important feature of this bacteriophage is that it does not kill its host cell. ये अपने host को kill नहीं करता है, so we can produce a huge number of progenies from a single bacterial cell. So therefore, large quantity of M13 bacteriophage can be easily harvested, along with our gene of interest. So this M13 vector is prepared for cloning by cutting its circular replication form DNA at a single site with the help of enzymes which are known as restriction enzymes. तो replication form के पास से जो restriction enzymes है वो यहाँ पे एक cut produce करते हैं for ligation of gene of interest. Then the cut is made in a polylinker region. एक specific region होता है इस genome के ऊपर, M13 के genome के ऊपर, that contains a series of closely placed recognition sites for restriction enzymes. तो यहाँ पे closely placed sites होती हैं for recognition by these endonucleases or restriction enzymes, but only one of each sites present in a vector. तो इनमें से सिर्फ एक ही साइट present होती है जहाँ से restriction endonucleases इसे cut करते हैं. Then the same restriction endonuclease will cut the DNA of interest. So a double-stranded foreign DNA जो हमारा gene of interest है उसे भी same restriction enzyme cut करता है. Fragment constructed by cleavage with the same restriction enzyme digest is then this gene of interest is ligated in this genome of bacteriophage. So this is ligated or pasted in this vector to form this rDNA. Then it is inserted by virus inside the bacterial cell or host cell, और वहाँ पे इसकी multiple copies ये produce करता है. And the foreign DNA can be inserted in two different orientations because the ends of both DNA molecule are same. तो दो orientations से ये इसमें insert कर सकता है foreign DNA को. So this is how we can use this M13 bacteriophage as a bacterial cloning vector for cloning gene of interest inside bacteria. The next important cloning factor is the Cosmid vector. It can comparatively carry a larger size fragment of DNA, gene of interest, as compared to the plasmid and phages. And this is a medium-sized vector, and it is made up of by the combination of two vectors. One is plasmid, and second one is the lambda phage. तो दो जो plasmid vectors हैं इनके combination से इसको बनाया था. So we can say this is a hybrid vector in which the origin of replication, selectable marker genes and cloning sites are obtained from plasmid, and the cohesive sites or cos sites are obtained from the lambda phage. So this is the combination of these two vectors. Basically, Cosmid vector is a medium-sized vector, and its cloning capacity is 35 to 45 kilobases. तो इस size का DNA insert ये clone कर सकता है. And this was first described by Collins in 1978, and having properties of plasmid vector and the bacteriophage vector, that is the lambda phage. तो इन दोनों की properties इसमें होती है. Here this ORI, that is origin of replication, then selectable markers, multiple cloning sites, these are obtained from this plasmid, and the cos sites or cohesive regions. So this cohesive region is obtained from lambda phage. About 200 base pair lambda phage sequences is cloned into the Cosmid vector. तो इतने sequences का जो DNA है lambda phage का उसे clone किया जाता है इस Cosmid vector में, and it may have one or two cos sites. इसमें एक या दो cos sites हो सकती हैं, cohesive sites. Cohesive sites are meant for attachment of जीन ऑफ इंटरेस्ट, तो इसी जगह पे हम जीन ऑफ इंटरेस्ट को इंसर्ट करते हैं. जनरली यूज फॉर द जीनोमिक लाइब्रेरी कंस्ट्रक्शन, जो लार्ज जीनोम डिसाइफरिंग के प्रोजेक्ट्स हैं उनमें इस वेक्टर को मोस्टली यूज किया जाता है. नेक्स्ट, हाउ we can ligate the gene of interest and how we can produce multiple copies of that gene of interest. So there are following steps which are involved in this. First is ligation of foreign DNA between the two cos sites. जो cos sites हैं, इन दो cos sites हैं, इनके बीच में यहाँ पे gene of interest को पहले ligate किया जाएगा with the help of DNA ligase. Then it helps in making concatenameric DNA. This concatenameric DNA is the DNA which contain multiple cloned sequences from different region of DNA. तो अलग अलग रीजन के जो सीक्वेंसेस है उसको एक ही जगह पे कॉपी कर लेता है ये, और वो जो डीएनए होता है दैट इज कॉल्ड concatenameric DNA. So it involves in vitro packaging of DNA into the phage head to form the matured phage particle. तो इसमें जो डीएनए होता है उसे हम पैकेज कर देते हैं फेज के हेड के अंदर, जैसे वो लगता है हमें कि एक मैच्योर वायरस पार्टिकल है. दैन दिस क्लोन डीएनए इंट्रोडक्शन इनटू द E.coli, जिस डीएनए को क्लोन करना है उसको फिर विद द हेल्प ऑफ दिस वायरस इट इज इंट्रोड्यूस्ड इनसाइड द होस्ट सेल दैट इज E.coli सेल बाय द प्रोसेस ऑफ ट्रांसडक्शन. एंड आफ्टर द एंट्री द कॉस्मिड आर मेंटेन्ड एज प्लाज्मिड. तो इन कॉस्मिड्स को प्लाज्मिड की तरह मेंटेन किया जाता है सेल में, एंड दीज आर मेंटेन्ड एज हाई कॉपी प्लाज्मिड. दे कैन प्रोड्यूस मल्टीपल कॉपीज ऑफ दिस cosmid और plasmid की तरह इसे मेंटेन किया जाता है. And one important application of this vector is constructing genomic library of organism with large genome size is that they have cloning capacity about twice as that of lambda vectors. तो जो ये lambda bacteriophages होते हैं उनसे डबल ये cloning capacity इसकी होती है. But there is one disadvantage that some cosmic clones are unstable on propagation in E.coli due to high copy number plasmid replication system. तो इस वजह से ये E.coli के अंदर कई बार प्रॉपरली फंक्शन नहीं कर पाता एंड बिकम्स अनस्टेबल. सो दिस इज ऑल अबाउट दिस cosmid vector. So this was all about for our today's discussion about cloning vectors which are used in case of E.coli. In next presentation I will discuss about the vectors which are used in case of yeast, plants and some mobile elements like we have transposons. Hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in comment section. Thanks for watching. Have a nice day.